नाही सुरू नाही वेबिनार सुरू झालं पण ते नाही झालं 5:30 ला आहे. रिचर्ड रॉबर्ट नोबल लॉरेट इन मेडिसिन अँड फिजियोलॉजी. पूर्वाला सांग.
Hello? होण्याला वेळ लागणार. आपण रॅन्डम टेस्टिंग करता आपण फक्त सिम्प्टोमॅटिक लोकांचे टेस्ट.
Richard Roberts Nobel Laureate in Medicine and Physiology.
Hello. Somebody is speaking in between. Please be quiet, and we'll begin sharp at 5:30. Who is there on the Zoom? He has joined.
Majority of customers face scarcity of FMCG items in Mumbai and lockdown.
आता तुम्ही diaper, sir?
Yes, ma'am.
Yes, begin.
Good evening to all the delegates from India, and good afternoon to our chief guest today. Greetings to all the dignitaries and participants. My name is Dr. Baliram Gaikwad. It's my honor, privilege, and the rare opportunity to host this session, The Path to the Nobel Prize, which I'm sure that many of us are attending for the first time in our lives. What makes this session a unique experience is the outstanding speaker for today's session. Our illustrious speaker in this session is none other than Professor Sir Richard Roberts, Nobel laureate in physiology of medicine in 1993. I invite Dr. Meenakshi Gurau, the President of International Board for Education, Research and Development, Mumbai, to introduce our speaker of this session. I request Dr. Meenakshi Gurau, ma'am, to please introduce our speaker of the session.
Thank you, Bairam. Good evening, everybody.
Meenakshi, ma'am.
Am I audible? Can you all hear me?
Yes, ma'am, we can.
Thank you. So thank you, Dr. Bairam. Good evening to one and all over here, honorable excellencies, respected dignitaries, and everyone on this platform. I'm profusely overjoyed to take this opportunity to introduce Sir. I'm very delighted to speak about Professor Sir Richard Roberts. I met Sir in December 2018 on one of the occasions, and since then I dreamt of having him with us so that we all can be inspired by his excellent thoughts. Sir Richard Roberts was born in Derby on September 6, 1943, and as a young pupil expressed an interest in logical problems and mathematics. At the age of 18, he was admitted to the University of Sheffield, where he studied chemistry, physics, and mathematics. 3 years later, he was accepted as a candidate for doctoral degree in organic chemistry. While examining the substances contained a tropical wood, he discovered new compounds and interrelationships which led to an interest in biology. Already a passionate reader, he began avidly reading about the molecular biology. After obtaining his PhD, Sir Robert took on the position as a postdoctoral fellow with Professor Jack Strominger in biological laboratories at Harvard University, USA. Thus, in 1969, a new period of life began for him as a scientist. A stay as a guest in Cambridge with Frederick Sanger, who in 1980 was awarded a Nobel Prize in Chemistry for the second time. He directed him to the technique of RNA sequencing, and he succeeded in exactly determining the individual nucleotide within some tRNA molecules, beginning a passion for nucleic acid and sequences. In 1972, Sir started working at Cold Spring Harbor Laboratory in New York. There, he discovered that the genes in the cells of higher organisms have a mosaic structure in which coding and non-coding sequences are interspersed, where in bacteria like E. coli, the genes are continuous. This result was achieved by Philip A. Sharp with his research team at MIT in Cambridge, MA in 1977. Both the scientists were jointly awarded the Nobel Prize in Medicine 1993 for their discoveries of split genes. Their discovery led to decisive process in many fields, including cancer research. Since 1992, Sir has been working as a research director at New England Biolabs, a molecular biology reagent company located in Beverly, Massachusetts. His current research focuses on DNA methylation in bacteria and the use of bioinformatics to discover new bacterial gene functions. He is currently leading a campaign that includes 131 Nobel laureates who supported that use of inherent safety of GMO techniques for improving plant varieties, especially those needed in the development of the countries. He was knighted in 2008. I know that all of us are very eager to know his path to the Nobel Prize, and me too. I wind up to my extended introduction about him and request Sir to bestow us with his words of wisdom. Over to Dr. Bairam to take the proceeding ahead. Thank you. Thank you very much, sir, for joining us on this session. Over to Bairam, Dr. Bairam.
Thank you very much. Yeah. Thank you very much, Meenakshi ma'am, for your wonderful introduction of our guest. Thank you very much, researchers. We may have attended myriad sessions. Hello.
Yes, please continue, sir.
Thank you. Thank you very much for, thank you very much, Dr. Meenakshi Gaurav ma'am. Ladies and gentlemen, as an academician, researchers, we may have attended myriad sessions in our life, but this session today is going to be the rarest of the rarest. Dear participants, let's get ready to welcome our distinguished speaker, Professor Sir Richard Roberts, Nobel Laureate. Sir, I request you to please enlighten us on this occasion.
Hello, can you hear me?
Yes, sir.
Okay. I'm going to share my screen to bring up the slides.
Absolutely.
Yes, sir.
Okay, that means that you have to stop sharing your screen.
Yes, sir. Please continue.
Okay, fine. So can you see that okay?
Yes,
Good.
sir.
Good, good. So you've already actually heard a rather precise summary of my talk from the earlier speaker, but I will go into a little more detail about it at this point. As was told previously, I was born in 1943 in Derby in England, and at that time Britain was at war with Germany, and the street on which I lived was actually rather a dangerous place because it was on the direct route for German bombers that were trying to bomb the Rolls-Royce factory just down the road. And as a result of it, half of my street was bombed, but fortunately I lived on the side that wasn't bombed. Now in 1947, my parents moved to Bath in England, and it was really in Bath that I built up all of my childhood memories, and it was there that I really started an interest in science. I was always very keen on puzzles. I like puzzles very much, and especially mathematical ones. I had a teacher at a junior school who used to give me puzzles to do at the end of the day, and I loved it. I thought this was great, and it wasn't until much later that I discovered I was the only person that he was doing this to. He didn't seem to have any interest in doing it to someone else. Anyway, that really started me thinking in general about puzzles, and I read a book called How I Became a Detective, and just a regular detective, a police detective, and I realized that this was something that I was absolutely fascinated by, and I thought perhaps I could become a detective. However, mathematics got in the way, and I became interested in mathematics and thought that my future lay with mathematics. But later, when I was about, I don't know, 12, 13, my father, mother and father bought me a chemistry set for Christmas. And it was one of those chemistry sets that are not of the sort you can get today, but it was one that actually had a lot of interesting chemicals in it. And I discovered that you could do many things with these chemicals, including make fireworks and explosives. And so I absolutely fell in love with chemistry at that time. I thought this was great. And my father was always very helpful, and he built a sort of a mini chemistry lab in our kitchen. And I did all sorts of experiments and spent a lot of time searching chemistry, researching chemistry, looking into chemistry, reading books about chemistry, and learning what I could do with this chemistry set. And I was very fortunate that my parents, while they didn't know chemistry themselves, they were not educated. My father and mother had left school early. Nevertheless, they encouraged me. They saw this was something that I loved to do, and so they encouraged me in all of this. I have to say, my father was not an especially smart man, but he was one of these people who worked really hard. And I think this was something that I learned from him and got from him. Maybe it's in the genes, maybe I inherited it. But I've always been a hard worker at whatever I've decided to pursue. My mother, on the other hand, was somewhat different. My mother was quite intellectual, although she never had a chance to learn how to benefit from that while she was at school. But nevertheless, she passed a love of learning and inquiry on to me. Now, after school, I did chemistry at school. I pursued chemistry at Sheffield University. And what happened at Sheffield was that for the first couple of years, I did the standard courses, chemistry, math, physics. And then in the second year, I also did some biochemistry, although I actually at the time found that rather boring. But at the end of that time, you take a degree, a pre-bachelor's examination to get some idea of how you're doing in terms of your final examinations after 3 years. I took those examinations and I did actually pretty well in them. And I was rather surprised because I must say that while I was at university, I spent a lot of time doing things that had absolutely nothing to do with learning chemistry. I learned to play bridge. I spent quite a lot of time doing things that were unusual, things that one normally wouldn't do as part of the regular curriculum. Did a certain amount of reading, made lots of friends, and so on. And so the fact that I hadn't really worked very hard for this examination at the end of the second year came as a bit of a surprise to me. I didn't think I would have done as well. And I was talking to my tutor, and he said to me, well, you know, your scores were such that with just a little bit more, you could get a first-class honors degree in chemistry. The grade at the end of the second year had been the grade at the end of the second year had been an upper second class degree. Hello, is someone trying to talk to me?
No, sir. Please
No?
continue.
Okay, so as a result of that, I decided that maybe if I did work hard during this third year, I would get the first class degree. And so for one of the first times in my life, I really studied hard. I previously, you know, pursued various paths, but I'd always been rather interested in lots of different things. So anyway, I worked really hard at chemistry during this third year, took the exam and got an upper second. And that decided me that all of this sort of forced learning and the standard way of learning was not for me. It was not a good thing to do. But nevertheless, as a result of that, I remained interested in chemistry. I applied to be a graduate student at Sheffield, and I was accepted by Professor Ollis, who was the head of the department at the time. He was an organic chemistry. And he was one of these interesting teachers who emphasized the use of problem solving when you're learning. So the typical chemistry lectures would involve some background material and then a series of questions where you had to solve problems in order to get the answer. And this made learning really fun for me. It was not the chore that it had been previously. So anyway, I went on, did chemistry. I was given a piece of wood from the Brazilian jungle and told to find out what was in it. Now, while I was in Sheffield, as a graduate student, I worked in a lab with Kazu Kurosawa, the gentleman shown here. And Kazu was really an amazing person. I learned more from him than I think I'd learned from anybody. He was one of these people who would explain to you what you should be doing, how to do it, and at the end of it, you would understand why you had done it. And as a result of this, pretty much all of the experiments that I did under his tutelage worked rather well. And as a result, at the end of the first year, I'd isolated a lot of rather interesting compounds from this piece of wood, including a rare biochemical intermediate in a reaction that led to a final product called a neoflavonoid. And my professor had predicted that this intermediate would exist. Other people had looked at it, but it had not been stable in the compounds they were looking at. But in my tree, this compound was stable. And so he was really happy. And the bottom line was, at the end of the first year, I really had enough material that I could have written my thesis. I did write several papers at the time. But in England, you have to spend 3 years doing your PhD. And so I had a lot of time on my hands. I devised a project myself for the second year that didn't work at all. But all the time I was with Kazu, he taught me many things, including chemistry and including how to play Go, the game shown at the bottom left. And in fact, after he left Sheffield to go back to Japan, we used to play Go by mail for quite a long time. And I still like games and puzzles very much. They're something that I enjoy greatly. But the other thing that happened was that I used to spend a lot of time in the library at Sheffield. And I read this book called The Thread of Life, which was written by John Kendrew, himself a Nobel Prize winner. And he was one of the first people to get involved in the field of molecular biology. And The Thread of Life was a history of molecular biology. And when I started reading this book, I knew a little about molecular biology, at least I knew what it was. But by the end of the book, I knew this was what I had to do with my life. I wanted to be a molecular biologist. And it was interesting because after I won the Nobel Prize, I wrote this in my Nobel autobiography. And John Kendrew wrote to me, and he said he was absolutely delighted to learn that his book had had this effect on me. He said it was the nicest thing that anybody had ever said about his book, because apparently the reviewers thought it was terrible. Fortunately, I hadn't read the reviews ahead of time, but for me, this was the book that changed my life. And I always recommend this to students, that when you're studying any subject, be sure to read things around the subject, read things that might intersect with it or might not. But very often you will find that there are things that you can read about or listen to, lectures you go to, that are more interesting and look like more fun than what you're doing at the moment. And if that is the case, never hesitate to change. Really, I think change is so important when you're growing up as a scientist, that you just do not know whether the subject you're studying at the moment is going to be the thing that will become the passion in your life. Now, after I finished my PhD, I applied to 6 places in order to do a postdoc, because I actually liked the academic life in England and thought I would become an academic. But in order to do so, you had to go abroad. And I applied to 6 labs that were doing molecular biology, but with a chemical bias so that they might think I had something to offer. And of the 6 people that I applied to, only one offered me a job. And 5 of them just turned me down flat. The person who offered me a job was Jack Strominger, shown here. Jack is really an incredible man. I had heard him speak when he came to Sheffield and described about 50 man-years of work in an hour and made it all sound very simple. And he talked about the synthesis of cell walls in bacteria, how they make the wall that surrounds the cell to keep it intact. When he accepted me, he was at Wisconsin. He was a professor at Wisconsin. And so I thought I would be going to Wisconsin in order to do my work. But a few months before I was scheduled to go, he wrote to me and said, could you delay coming for 3 months? Because I'm just moving to Harvard as a professor. He'd just been appointed a professor there. So I did that and then moved to Harvard. Quite unexpectedly, it was not something I was expecting to do. He had just arrived at Harvard and at the time had decided to change fields. He started working on HLA antigens and more eukaryotic things. But there were a few projects left over from his bacterial cell wall days, and he gave me one of those to work on. I was actually an understudy, as it were, to a more senior postdoc who was just finishing up as a postdoc, and he had started on this project of trying to determine the sequence of a transfer RNA from Staphylococcus epidermidis. He told me everything he knew, got me going on the method that he was using to determine the sequence, which was a rather old-fashioned method. It was one that was devised by Robert Holley, who had won a Nobel Prize, and it was not what I would call the most effective way of sequencing RNA at the time. As soon as Tom Stewart, the postdoc, left, I started reading more about this area and learned that Fred Sanger at the MRC lab in Cambridge had developed what looked like a much better method of sequencing tRNAs. I switched my approach and started on the approach that would allow me to use Fred Sanger's methods. And I was fortunate enough to be invited. I wrote to Fred Sanger and asked if I could go to his lab to learn the methodology. And I was fortunate that he said yes. And so I ended up going to the MRC lab in Cambridge in 1970 to work with Fred Sanger. Fred Sanger, shown here on the right of the photograph. He had won the Nobel Prize in chemistry for discovering how to sequence proteins. Later, he won the Nobel Prize in chemistry again, one of the very few people to win 2 Nobel Prizes. And this was for determining DNA sequencing. He could, of course, in the meantime, have won it for developing the methods for RNA sequencing, but I guess they thought that 3 prizes might be too much. Anyway, he was a quite remarkable man. I remember arriving in his lab and seeing this little man who was busy washing the dishes at the time, and it was a Sunday morning, and I thought it must be the janitor who had come in and so on. And I introduced myself and said, I'm Rich Roberts. I'm come here to work with Fred Sanger. And he turned and smiled, shook my hand and said, I'm Fred Sanger. This was my introduction to him. And I think Fred was always one of my great heroes, perhaps the greatest hero that I knew in molecular biology. He was a quite remarkable man, very modest, extremely smart, but he would never let you know how smart he was. So that led me to an interest in sequencing, and that led to the next stage of my life eventually. In 1972, I had to start looking for a job. I'd been at Harvard for 4 years, and I was offered 2 jobs. One was a job at Harvard itself in the biology department, but the other was from Jim Watson, who offered me a position at Cold Spring Harbor Laboratory, and he wanted me to sequence the DNA of SV40. I had a very unusual interaction with him. Mark Potashny had told me that Jim was interested in hiring someone at Cold Spring Harbor to sequence DNA, and I might be the person that would go on. And he said, Jim will come and contact you. However, Jim never came, and Mark said, well, Jim doesn't actually know who you are, and perhaps you could go and introduce yourself. I went to his office, knocked on the door. He said, come in. I must have been in his office for no more than 10 minutes, and at the end of which he offered me this position at Cold Spring Harbor. And I figured he must I talked to Jack Strominger or talked to someone about me who knew me rather well, but it turned out that it was Mark Potashny who had recommended me for the job, and that led to this offer. Now, Cold Spring Harbor was a very interesting place at the time, and I had a really very nice time while I was there, although I have to say I got into trouble fairly early on. Jim hired me specifically to sequence SV40 DNA. Now, at the time, SV40 was a small virus, a very small one. It was one that a number of molecular biologists had chosen to study, but there were two other groups already sequencing it. And I've never been one to believe that competition is a good thing, especially in biology, where there are so many things that we don't know. It is always possible to find something new to do that is not actively competing with other people. And so when I got to Cold Spring Harbor, I thought rather than just sequence SV40, I would take advantage of something else. And that was the fact that I had heard a lecture by Dan Nathans, the person at the top left of this picture, who later went on to win the Nobel Prize. But I'd heard him talk at Harvard when I was a student. He'd gone over to the medical school. I went over, listened to him, and he described some enzymes called restriction enzymes. And it was quite amazing to me that here were a set of enzymes that were going to be very, very useful in the sequencing field. Now, when I got down to Cold Spring Harbor, I immediately started looking at one of these enzymes, in particular, the enzyme that Ham Smith, the person at the top bottom left of this photograph, had discovered. It was called endonuclease R, and it was able to take SV40 and to cut it into small pieces. And I thought these small pieces might be a good substrate on which to learn how to sequence DNA. At the time, methods had been developed for protein sequencing and RNA sequencing by Fred Sanger, but there were no methods for DNA sequencing other than to copy it into RNA and then sequence the RNA, which was what the two groups working on SV40 were doing. So I thought, well, this restriction enzyme gave me a small piece of DNA to work with, and perhaps I could work out some methods myself that might enable me to learn how to sequence DNA. However, the methods that I tried to develop didn't work terribly well, but we also started looking for other restriction enzymes other than this endonuclease R. I always figure if there's one, there's going to be more. And we pretty soon discovered that almost everywhere we looked in bacteria and archaea, there were restriction enzymes. And so these became a passion of their own, because it turned out that these enzymes were very, very useful in many aspects of molecular biology, not just sequencing. By the time that we had really stopped looking for new ones, we had found 75 of the first 100 known type 2 restriction enzymes in my lab at Cold Spring Harbor. At the time, these were not commercially available, and so people used to come to my lab in order to see if we had an enzyme that would cut their favorite piece of DNA. It occurred to me that here was a commercial opportunity because people didn't want to make their own, and a company that could make a large number of these would be very useful to the molecular biology community. I talked to Jim Watson about this and suggested that Cold Spring Harbor should set up a company to make these enzymes and then use the profits to support the research at Cold Spring Harbor. Jim was not impressed. He did not think that you could make money doing this, and he was also attuned to the more general atmosphere in molecular biology that making money was somehow a little bit dirty and was not something that serious academics would get involved in. And so it was that I ran into another man called Don Cohn, who got New England Biolabs going. He had started New England Biolabs after having been denied tenure at Harvard. He'd been an associate professor in the biology department at Harvard. He was denied tenure, and he decided that academy was not for him, at least not traditional academic life, and that he would start a company. He did that in about 1972, 73, and went on to do a number of things that looked as though they could be commercially successful, but in fact, they were not terribly successful. The most important thing he did was to get involved in doing pregnancy testing for ladies. When he heard about restriction enzymes, he thought that this might be something good to do. And he and I had this same idea that making restriction enzymes and using the profits to support research would be a very good thing to do. And so I signed on as his chief consultant. We sent the enzymes to him, told him how to make them, and then New England Biolabs got going. This turned out to be incredibly successful from day one. In the first 12 months, they sold about $200,000 worth of these enzymes. And this was back in 1975, and when $200,000 was really a great deal of money. New England Biolabs has gone on to be a very successful organization. I moved there permanently in 1992 as research director, and then became their chief scientific officer. I still work there. We have been doing a great deal of interesting work over the time. We have more than 100 people at the company doing research, most of it very basic. And recently, quite a bit has turned to studying coronavirus, and we've been developing tests for coronaviruses. Many of the reagents we sell are being used in tests for coronavirus. And so it turns out the coronavirus pandemic has been extremely good for New England Biolabs. We were making reagents that were needed. Now, to get back to Cold Spring Harbor, postdocs, I started having postdocs come into my lab. And one of these postdocs in 1974, Richard Gelinas, was someone that I'd met at Harvard. He'd been a graduate student when I was at Harvard, and he applied to come and be a postdoc. And we got involved in a project that was going to turn out to be absolutely important in molecular biology. However, we started it, as usual, by having a very specific goal in mind. We thought it might be possible to identify DNA promoter regions, the regions at the 5' ends of genes that told the RNA polymerase where it should start making RNA. We were going to do this on adenovirus 2 DNA, which is a small virus and one that works well in the laboratory, easy to deal with. And basically, the question was, do eukaryotic promoters, because adenovirus was a eukaryotic virus, do they look like promoters in prokaryotes, in bacteria? We got going. We used a technique that I devised that had never been used before to study this particular kind of problem. And things went extremely well. However, we initially started looking at early promoters in adenovirus, those that were employed as soon as the virus infected a cell. But it turned out we had trouble getting enough material. And so we then turned our attention to late promoters, where the entire transcription apparatus in adenovirus is making adenovirus messenger RNA. This turned out to be much better, but we ended up getting some very strange results. Oh, actually, let me go back to the previous slide. One of the things we found was that the late promoters, which were starting around position 20, a little beyond position 20 on this map, that this promoter, these RNAs, all had the same 5' end of their messenger RNA. And it was as though what was going on was that there was some piece of RNA that might be priming the synthesis of these messenger RNAs. And we had an idea what it might be, because there was a small RNA encoded by RNA polymerase, decoded by RNA polymerase 3 that we thought might be the primer for synthesis. It turned out, of course, that this was not correct, but rather what was going on was that there was a very long RNA transcript being made that went from position 20 all the way through pretty much to the end of the viral genome. This led to the discovery of RNA splicing. It was called split genes, and led to the notion that in higher organisms, instead of the genes being single pieces of DNA that had a defined start and end with no gaps in them, that in eukaryotes, the opposite was true. What happened in eukaryotes was that you made very long pieces of RNA, and these were then cut and spliced to make the messenger RNA that looked like a typical bacterial messenger RNA. Now, to do the key experiment that showed what was going on, I came up with this electron microscope experiment that is illustrated here on the right of the slide. Basically, we made an R loop between a messenger RNA from adenovirus, and we anticipated that the 5' end of it would come from somewhere other than the main coding region. When we did this, we came up with a very nice experiment. Now, neither Richard nor I were electron microscopists, and so we went and talked to our colleagues, Louise Chow and Tom Broker, who worked just down the hall. They did this experiment for us. We devised the experiment on a Saturday morning. Richard worked on Sunday to make the reagents, and on Monday had the reagents. We gave them to Louise, and she did the key experiment on a Tuesday morning. And at that point, this picture that I had drawn on the blackboard on Saturday morning, we saw it in the electron microscope. And at that point, RNA splicing was discovered. There was just one interesting problem at the time, was that instead of there being just a single piece of RNA up here at the 5' end, there were actually two separate pieces, but they were joined by a loop. And it was this that led to the idea of RNA splicing. The work was published in Cell in 1977 and was called an amazing sequence arrangement at the 5' ends of adenovirus 2 messenger RNA. I have to tell you a little story about this. And when we sent it in, went to Cell, the reviewers wrote back, 3 reviewers, all of whom said they thought amazing was not an appropriate word to use in the title of a scientific paper. And so I called Ben Lewin, who was the editor of Cell at the time, and we had a little conversation about some of the other points the reviewers had raised. And during the conversation, I asked him, when you first heard about this, what did you think? He replied, I thought it was amazing. I said, I rest my case. We want to use the word amazing in the title of this paper. And he agreed. So apparently I'm told this is the first time that amazing was used in a scientific paper. Now, just what are split genes? This is a simplified diagram. At the top here is a typical gene, just here, that is found in a bacteria. Single piece of DNA runs from the left here to the right, and you read the bases 3 at a time. They are codons in the mRNA that make protein. However, in higher organisms, the genes are split into pieces as shown at the bottom. You make a single piece of RNA that copies this entire region, and that becomes a pre-messenger RNA. And it is that messenger RNA that is cut and spliced to bring the final mRNA. And that mRNA looks exactly like a bacterial mRNA that goes to the ribosome, is translated 3 bases at a time to produce a mature protein. So this was a fundamental discovery in molecular biology. And one of the nice things was that it was not someone that anyone had predicted. Very often you make a discovery and you discover someone else predicted it ahead of time, and all you do is confirm their theory. in this case, this had not been predicted. And so that was really very nice. That led to the Nobel Prize in Physiology or Medicine in 1993, which I shared with Phil Sharp, who had made this same discovery independently while working at MIT. It's interesting that I had actually gone and talked to Phil a few months before we made the final discovery and talked about these unusual results we'd been getting. But he didn't mention at the time that he had had, or one of his postdocs had had some similar results, but it turned out he hadn't really believed her. And so it was only later, again, independently of anything that we did, that he came up with the same solution to the problem. Now, I have to say, winning a Nobel Prize is really quite an extraordinary thing. It's something that I recommend to everybody. One of the things about Sweden is that they really know how to throw a party. And so the Nobel Prize ceremonies and all of the accompanying parties that take place are really quite extraordinary. Among other things, you get to meet the King of Sweden, King and Queen of Sweden, go to a banquet, and the whole thing is just wonderful. Very, very exciting experience. Now, when you go to Stockholm to pick up a Nobel Prize, one of the things you have to do is to give a talk. And that talk is normally about the work that you did that led to the Nobel Prize. But in my case, I had actually stopped working in the field many years previously. However, we had discovered something else in the meantime. In fact, we had discovered it just 1 month before I had to go to Stockholm and give the Nobel Prize speech. And this was a discovery that was made with Xiaodong Cheng, who was then a postdoc at Cold Spring Harbor, but worked in the electron microscopy lab. Not the electron, the X-ray crystallography lab, I'm sorry. He turns out to be a wonderful crystallographer. And we had been working on DNA methylases, and in particular, we wanted to get a structure of one of them. We had given this compound, HHA1 methylase, to Xiaodong, and he fortunately was able to get a crystal structure. And what that crystal structure showed was that when the protein bound to DNA, instead of just binding and keeping the DNA structure intact, as it normally does, as most DNA binding proteins do, in this case, it flipped the base that it was going to methylate right out of the DNA. And it was when it was in this position in the protein that it could be properly methylated. Turns out that this was the first demonstration of base flipping, again, something that no one had predicted. And it now turns out to be very common for proteins that do chemistry on DNA to use this base flipping mechanism in order to get the base into the protein where the chemistry can be done. So this was an exciting discovery, and this formed the basis for my Nobel talk. And I always feel if you have to find a venue where you can describe an interesting discovery, Stockholm is obviously a good place to do it. Now, of course, once you win a Nobel Prize, many honors follow. I've had several buildings named after me, including one at the University of Sheffield, one of the labs of the chemistry department, and also at my school back in Bath, where the chemistry department building is now named after me. In 2008, I was knighted by Prince Charles at Buckingham Palace. At the bottom left is my family, my daughter on the left, Amanda, my wife, Jean, standing next to me, and my son, Chris. And this was a very wonderful, very exciting event to go there. I've also had a number of honorary doctorates of one sort and another conferred on me. And one of the more interesting ones was the honorary doctorate of medicine that I got at the University of Uppsala. And this was the first honor that I received before the Nobel Prize. In fact, the Nobel Prize was the first prize that I ever won. Now, one of the things that's happened since I won the prize, is that I made an interesting discovery. I discovered that before I won the prize, I would often talk to people and say what I considered words of wisdom, and people would listen politely, but rarely would they act. After I won the Nobel Prize, I discovered that people started listening to me, and in some cases, action followed. And this was quite a revelation. I decided that perhaps one thing I could do as a Nobel laureate was to organize some of my Nobel friends to try to get some things done. One of the first things that I got involved with was calling for open access to the scientific literature. To this day, I'm a very big advocate of open access. And in fact, I won't publish any papers with my name on them in journals that are not open access. After that, which was successful in the early days in getting people started thinking that this was a good idea, the Public Library of Science journals followed this model, and now many journals are following this model. But unfortunately, there are lots that are not, that are still interested in trying to make money and trying to restrict access to the scientific literature. But there is growing concern at the moment that open access is a good thing. The next thing that I got involved with was serendipitous. It was something I learned by reading, that there were some nurses who'd been put in jail in Libya after having been accused of spreading HIV among children in the Benghazi Children's Hospital. They were accused of doing it deliberately. They were tortured to a point where one or two of them actually admitted that they had done it deliberately. And then they were being sentenced to death in a Libyan court. I'd heard about this early on, but the evidence was so strong that it was clear they didn't do it deliberately, and that I decided perhaps the Nobel laureates could help with this too. To cut a long story short, we were able to gain the release of the Bulgarian nurses from jail in Libya. I went over to Tripoli. I met with Gaddafi's son, Saif al-Islam, and he assured me at the time that they knew that they were not guilty of this crime, and they got released. But here, the Nobel laureates played a major role in getting not just the scientists behind it, but also a number of politicians in Europe. At the moment, I'm involved in a big way in another campaign promoting the safety of GMOs and asking the anti-GMO activists to start telling the truth about the science and not to keep pretending that these are dangerous. Rather, very much the opposite. They are good for humanity. We now have 155 Nobel laureates who've written an open letter to Greenpeace and to the UN ambassadors, urging an acknowledgement that GMO technology is safe and should be supported, particularly for the case of the developing world. And of course, India is a prime example. BT cotton in India has been incredibly successful, and yet the Indian government still refuses to allow the free growth of GMOs. And in my view, that's a big mistake. Now, in these closing minutes, I want to talk about the importance of luck. I think one of the reasons that I've been successful in my life is because I learned the importance of luck very early on. When I was 17, I stopped going to school. I got bored at school, and I spent a lot of time going down to the local YMCA and playing billiards and snooker. I became rather good at billiards and at snooker. I, in fact, became the West of England junior champion at snooker. But I got caught for not going to school, and I was threatened with expulsion. And so I looked around to see what I might do as an alternative career. I was playing snooker a great deal. I actually went to audition to be a professional snooker player. But I learned one of the most important lessons of my life while playing snooker. One day I was playing in the final of an important match, and I had a lucky break. I tried to do one thing, but instead something else happened, and nevertheless it was a scoring shot and allowed me to continue the game. However, the next shot I played rather carelessly and lost that advantage. And afterwards, an old man, a very old gentleman, came up to me and he said, "You know, when you made that lucky shot, the most important thing you have to learn is that when luck comes your way, you have to take advantage of it. And that means concentrating twice as hard on the next shot and not feeling guilty because you had the luck. Because everybody has luck in life, everybody. If you don't take advantage of it, then you're something of a fool. You should take advantage of the luck that comes your way." The second lucky break was working with Kazu Kurosawa. He was a postdoc who taught me more about chemistry than anybody else I know. And I've always looked to find good mentors, to find people who will give me good advice as I go through life. I went to Harvard instead of Wisconsin. That was another incredibly lucky break. At Harvard, I was exposed to a wide variety of people that I would never otherwise have met. Another piece of luck was looking for eukaryotic promoters in adenovirus. We did not start this project thinking we were going to make a big discovery. We thought we would just be making a small discovery about what eukaryotic promoters looked like sequence-wise. Were they the same or were they different from bacterial promoters? Instead, we discovered that some experiments we were doing didn't quite work as we expected. And so instead of trying another experiment, we looked to see why our experiments weren't working. And I always think that this is a very good thing for young scientists to know that failure is actually a good thing. When things work all the time, you don't learn very much. But when they fail and you can work out why they failed, that is a learning experience beyond any that I know. In fact, if you look at many people who are now running big companies, they started small companies that failed, but they learned along the way and eventually found out how to run a company properly. The next one is studying bacterial RM systems. Who would have guessed that we would have discovered base flipping as a result of doing this? In fact, the RM systems have provided a good backdrop for my entire life in science. And the last one is probably the luckiest thing that ever happened to me. You probably all remember the planes that went into the Twin Towers in New York in 2001. The first of those planes I was booked on until 2 weeks before it took off. I was scheduled to go and talk at a meeting in California and was flying to LA. However, the meeting was moved 1 day earlier. And as a result, I had to cancel my reservation on the plane that went into the Twin Tower, and I took that plane 1 day earlier. You can imagine my surprise when I woke up in California on the morning of 9/11 to discover that this plane that I was supposed to be on had crashed into the Twin Towers. I think for me, this demonstrates just how lucky I've been throughout my entire life. I would like to acknowledge the people who helped me over the years, Richard Gelinas, Louise Chow, and Tom Broker, who really made the discovery of splicing possible. Saida Zain and Dan Klessig were very important too. Saida was a postdoc in my lab, and she did some key experiments looking at adeno-sp40 hybrids. that really made it clear that there was something about eukaryotic transcription that was very different from what was going on in bacteria. Dan Klessig was a graduate student in a neighboring lab, and he worked very hard studying what went on in fiber messenger RNA in adenovirus. And of course, the many lab members, colleagues, collaborators, and friends over the 50 years that I have been practicing science. And finally, my admin assistant, Karen Otto, who prepared most of the slides that you have seen. And of course, I thank you for listening, and I'll be glad to take any questions you might have.
Any questions, please? Audience, you can send your questions in chat box or question answer, please.
Baliram, Dr. Baliram, please check
Yes, sir.
chat box, and
Yes, ma'am.
if there are any questions, you can ask.
Absolutely. Yeah.
Okay, so maybe I'll read the question, shall I? So the question is, what motivated me in life? Well, I think I have an insatiable curiosity. I always
Yes,
want
sir.
to know why things are the way they are. I like to, especially now, look at sequences, look at genes, and ask, what do they do? You know, it's remarkable in biology that even for the best studied organism, E. coli, still something like 20 to 30% of the genes, even some that we think we know what they do, we really don't know what they do. If we look at any new bacterium, typically many more than half the genes, we can't even make a good guess about what they're doing. And so there is so much that we don't know in biology, and I would like to find out more. My big hope is that before I die, we will actually work out how a bacterium works, just one bacterium. Okay, the next question is, what message should I give to budding researchers? I think that there are actually several messages. One is, find something that you're really passionate about. Don't get involved in something that you only care peripherally about, but make sure you make a career working on something that you're really passionate about. And the real significance of that is that if you're passionate about something, work just becomes a hobby. I don't really think that I work. I consider everything I do just to be my hobby. And so I do it morning, noon, and night, and love every minute of it. The other thing I would say is, don't be afraid of failure. Very often, when you do an experiment and fails, it's because nature is trying to tell you something. That what you thought was important, or what you thought was true, really isn't true. And this sort of thing happens all the time. In biology, in physics, in chemistry, very often things don't work because nature is trying to tell us something. I always see failure as an opportunity for discovery, not as something that one should worry about. Okay, so there are a number of questions that popped up. I think the moderator has to decide which one they want to ask. Yes, are you going to do that? Because now the questions have disappeared and I can't remember most of them. Okay, well, the one question I do remember, oh, what's this? What is the significance of DNA methylation? Is it the same in bacteria and eukaryotes? And the answer to that is very simple. No, it's not. In eukaryotes, methylation takes place during replication, and very often is just copying existing methylation marks. It seems that in eukaryotes, methylation has many functions. In some cases, it is a way of controlling transcription. In others, it's a way of passing marks from a parent into an offspring gene. And there are also many aspects of methylation, of epigenetic methylation in eukaryotes that we still don't understand properly. Same is also true to some extent in bacteria, but in bacteria, methylation is used by bacteria to protect against restriction enzymes, and restriction modification systems are an important defense mechanism to stop bacteriophage from infecting bacteria. We also know that there are other uses of methylation in bacteria. So for instance, in E. coli, DAM methylation, which is methylation of GATC sites, is something that is important in replication, in originating replication, and also in DNA repair, in marking which strand should be used as the template strand for repair. In Colobacter, we know that methylation is also involved in cell cycle modulation. In Clostridium, we know that methylation of a specific sequence is an important marker for sporulation, and that if you knock out the gene for a Clostridium difficile methylase, it can no longer sporulate. And this would make that methylase actually a very good drug target. There are many other orphan methylases in bacteria, and we don't understand what they do. Again, this is why I'm interested in this field, because there is lots still to discover. I think someone is trying to say something with their microphone muted.
Hello, sir.
Hello?
This is Principal Thakur from Panvel, Mumbai.
Okay.
Yeah, sir, what is your message in the present scenario of COVID-19 pandemic, basically for the researchers?
Well, you know, the current coronavirus pandemic is turning out to be very interesting. I think it's shown in many cases that political leaders are not as good as one might wish, that many political leaders, especially here in the US, have done an awful job of protecting the public and passing out good public health messages. But it's also shown that some leaders are very good. The Prime Minister of Taiwan has done very well. The Prime Minister of New Zealand did very well. So it's shown, I think, just how good or bad many political leaders are. From a scientific point of view, I think it has mobilized the community in ways that one might not have anticipated. A lot of people are now working on coronavirus, including some at New England Biolabs, where I work, that previously would not have considered working on coronaviruses or on any RNA viruses. Many people have been devising novel tests that one might use. Modern vaccine development may well come out as a big winner from this. I particularly am thinking about Moderna, who are trying to use messenger RNA as a way of vaccinating people, of delivering the proteins that are necessary to produce antigens. If their approach works, that is an approach that can be used very widely. It can be deployed very quickly, and it may herald a brand new realm of vaccine production. The one thing I would say on vaccine production is that if we are ever to develop these vaccines more quickly, that we need to take a step that we've not been prepared to take in the past, and that is to actually do experimental tests of vaccines on human volunteers. And coronavirus is perhaps a very good example of where we might do that. You know, when you do experimental tests on monkeys, you give them the vaccine and then you give them the virus. And we could do the same with humans if there were volunteers. And for the Oxford vaccine, I'm told that there are more than 3,000 people who've already volunteered to serve in this capacity. And in the case of human volunteers for coronavirus, we know that, for instance, you would not pick volunteers who had underlying genetic conditions. You would not pick people who were very old and not very young. But there is a fairly wide age gap where it seems that even if you do get infected, the virus doesn't kill you. And so such people would be wonderful volunteers to find out if vaccines absolutely work. You would need to pick the people fairly carefully, but you wouldn't need the vast numbers of people that we currently treat and use in tests of vaccines. So I think that is something that is on the table, and we will hear very soon whether that is going to be successful or not. There are many other aspects of coronaviruses, how they work, how the infection proceeds, whether or not people who are asymptomatic are always passing the virus on, or whether sometimes they're not, and so on. So I think it's been a boon for research, actually. I did notice one question that went past asking whether I was an atheist or a theist. I'm a confirmed atheist. And I actually practice my atheism by trying to convince everybody else that they should be atheists also.
The audience, any more questions? Okay, thank you very much, sir.
Okay.
Thank you very much for your remarkable guidance to us. We were able to visualize the crystal path to the Nobel Prize, the journey of your research, rigorous academic benchmarks, and the challenges you faced in your journey as a researcher, and the honors followed after the unimaginable persistent hard work. So it was a very inspiring session, and many scholars and the young researchers witnessed this session. They must have been highly motivated by your speech. Sir, Pulitzer Prize winner Robert Frost, who chose to shift from USA to UK for some time, and later in the poem, The Road Not Taken, he wrote, "I shall be telling this with a sigh. Somewhere ages and ages hence, two roads diverge in a wood, and I took the one less traveled by, and that has made all the difference." So through your speech, we were able to see how that change has made the difference. So we wholeheartedly thank you so much for joining here with us on this Zoom platform in International Conference on Empirical and Theoretical Research. We appreciate your time with us, and now I invite Dr. Bhushan Langi, the Secretary, International Board for Education Research and Development, to offer the official vote of thanks to you, sir. Dr. Bhushan Langi. Dr. Bhushan.
Yes, sir. Just a minute. Thank you, sir. Good evening, all. We have just listened to the powerful and thought-provoking lecture by our eminent guest, Nobel laureate, Professor Sir Richard Roberts. It is my pride privilege to extend my heartiest vote of thanks to you, sir, for accepting our invitation and gracing this occasion. Your presence today in this function has immensely enhanced its importance. We are extremely grateful to you, sir, for your insightful address. We are also very grateful to Honorable Dr. Mukund Sorgade, President, Think Pharma and CSO, MVRC USA, without whom we would have not enjoyed such enlightening talk. My deep sense of gratitude to each and everybody on this platform. Thank you very much.
Thank you.
Thank you.
The session is over. Mahesh sir.
Ganesham, एक मिनिट थांब फक्त, एकच मिनिट. Everybody is logging off. Baliram,
Participants
Baliram
can
open karo.
leave now. All
Okay.
participants, panelists can leave the meeting now.
Yes, yes, yes. Madam,
I
meeting.
thank everybody for
Should
joining
we
the session.
should we close the meeting, ma'am?
Yeah.
Please close the meeting. Thank you very much, everybody, for joining this particular lecture. Thank you on the behalf of the iBird. Thank you very much.
Thank you, sir.